Good day everyone. Today we'll be discussing DNA profiling or DNA fingerprinting. So what is a DNA profiling? So it is a technique used by scientists to distinguish between individuals of the same species using only samples of their DNA. So who invented it? So the process of DNA fingerprinting was invented by Alec Jeffreys at University of Leicester in 1985. So he is knighted as the most excellent order of the British Empire in 1994. So that's Alex Rees. So what are the stages of DNA profiling? So there are four stages of DNA profiling. The first one, cells are broken down so that the DNA will be released in that's inside of the cell. So if only small amount of DNA is available, pwedeng gamitin is or pwede siyang i-amplify using the polymerase chain reaction. So the second step, the DNA is cut into fragments using restriction enzymes. So each restriction enzymes cut DNA at a specific base sequence. So ang tawag doon sa mga nakat na DNA is restriction fragments. So this yields thousands of restriction fragments of all different sizes because the base sequences being cut may be far apart or close together. So pwedeng maging long fragment or short fragment yung magiging restriction fragment. So the third step, the fragments are separated on a basis of size using the process of gel electrophoresis. So ang ginagawa doon is yung mga fragments nandun sa mga tubes na yun, i-inject mo dun sa well. Itong well na to para siyang may gelatin sa taas, dun mo itutusok, kaya nga siya tinawag na gel electrophoresis. Tapos lalagyan mo siya ng electric current para mag-move yung mga samples mo na nasa tube. So, ang DNA daw is negatively charged, so it is attracted to the positive end of the gel. So, in-inject mo siya dun sa negatively charged na part nung gel. So, ang tendency nung DNA is pumunta siya sa positive side. Bakit? Di ba pag sinabi is opposite attract. So, kung yung DNA is negatively charged, pupunta siya sa positively charged. So, dito, the shorter the DNA fragments, mas mabilis yung pag-migrate niya papunta sa positively charged area dun sa gel electrophoresis kaysa sa mga longer fragments. So, DNA is separated on basis of size. So, a radioactive material is added which combines with the DNA fragments to produce a fluorescent image. Kasi hindi mo yan makikita nang walang kumbaga pangkulay dun sa mismong agarose gel or yung pinaglagyan mo nung sample. So a photographic copy of the DNA bands is obtained. So yan yung magiging itsura niya. So stage 4, the pattern of the fragment distribution is then analyzed. So DNA profiling is used to solve crimes and medical problems. So crime forensic science is the use of scientific knowledge in legal situations. So DNA profile of each individual is highly specific. So the chances of two people having exactly the same DNA profile is 30 million to 1. So except for identical twins. So what are the biological materials used for DNA profiling? We have blood, hair, saliva, semen, body tissue cells. So DNA samples have been obtained from vaginal cells transferred to the outside of a condom during sexual intercourse. So DNA profiling can solve crimes. So the pattern of the DNA profile is then compared with those of the victim and the suspect. So if the profile matches the suspect, it provides strong evidence that the suspect was present at the crime scene. But it does not prove that they committed the crime. So if the profile doesn't match the suspect, then the suspect may be eliminated from the jury. So example, a violent murder occurred. The forensic team retrieved a blood sample from the crime scene. They prepared DNA profiles of the blood sample and the victim and the suspect as follows. So was the suspect at the crime scene? So based dyan, malalaman mo na yes, yung suspect is nandun sa crime scene. Bakit? Kasi yung pattern nung suspect profile at nung blood sample from the crime scene ay pareho. So masasabi mo na yung suspect ay nasa crime scene. So we also have solving medical problems para saan? Paternity suit, inheritance cases, and immigration cases. So example, a paternity test. By comparing the DNA profile of a mother and her child, it is possible to identify fragments in the child which are absent from the mother and must therefore have been inherited from the biological father. So is the man the father of the child? Makikita nyo yung mother at child, mostly yung patterns nila is nagkakapare-pareho. Pero dito, dun sa man or dun sa father, sobrang wala siyang pinagkapare-pareho. So, may possibility na hindi yung man yung father nung child. So, famous cases in 2002, Elizabeth Hurley used DNA profiling to prove that Steve Bing was the father of her job, Damien. So, another famous case is Colin Pitchfork was the first criminal caught based on DNA fingerprinting evidence. So, he was arrested in 1986 for the rape and murder of two girls and was sentenced in 1980. So, we also have Joseph Mengele. He is known as the Angel of Death, a notorious member of the team of doctors who are performing deadly human experiments. So he drowned while swimming in the Brazilian coast in 1979. His remains were positively identified using forensic examinations in 1985. Lastly, O.J. Simpson was cleared of a double murder charge in 1994, which relied heavily on DNA evidence. So this case highlighted lab difficulties.